I'm John McCamant with BioInvest, and I'm here today with Dr. Dan Teper with Immune Pharmaceuticals, and we're going to discuss his in-licensed phase 1 molecule that will deliver potentially exciting phase 2 data in 2016.
Nice to be here, John.
Could you tell us a little bit about your company, and then specifically some of the catalysts for 2016, and then the last question, or you can put this in your biomarker, which I don't think has been discussed in any of our previous interviews today, Dan.
Sure. So Immune Pharmaceutical, as the name indicates, is an immunology company. Everybody focuses on immuno-oncology. We do have an early immuno-oncology pipeline, but really our primary focus and what's important for 2016 is immunoinflammation. And the lead drug that we have in immunoinflammation is called bertelimumab. It was originally developed by CAT in the UK. That's the company that developed the #1 drug in the world, Humira, which is used for, I believe, 13 different inflammatory conditions.
Impressive. So going forward, what is the trial we're probably looking for in 2016 as investors to gauge whether this drug candidate is going to be efficacious?
So our antibody targets a pro-inflammatory protein called eotaxin-1. And that protein, eotaxin-1, is also a biomarker of disease severity in a number of different conditions. So it's personalized medicine applied to immunoinflammation the same way personalized medicine has been applied to cancer, starting with Herceptin, where basically women with breast cancer that overexpress HER2 are candidates for treatment, and those who don't have HER2 then are candidates for other treatments. So similarly, with bertelimumab, we're selecting patients based on the overexpression of eotaxin-1, which can be done relatively easily, and we are developing a companion diagnostic to help physicians select those patients. So in 2016, our focus is on gastroenterology on the one hand and on dermatology on the other hand. So in gastroenterology, the lead indications are Crohn's disease and ulcerative colitis, also known as IBD, inflammatory bowel disease. Our trial, which has started enrolling patients, so we have patients into the trial, is a 42-patient placebo-controlled, double-blind trials of bertelimumab versus placebo on the background of corticosteroids. So it's a classical trial at that stage of development in a phase 2. It's important to remind everybody that CAT has conducted phase 1 clinical trials that have shown initial safety of bertelimumab, but also have demonstrated biological activity of the antibody in humans. So this is not the first time that we're administering the drug to patients. So this trial will allow us to determine whether bertelimumab reduces the symptoms of ulcerative colitis and also has disease-modifying activity, at least in short-term use. There's an induction treatment and maintenance treatment. This is an induction treatment trial. So it's essentially the same trials that Remicade, Humira, and more recently Entyvio, vedolizumab from Takeda, have conducted. It was designed with some of the top opinion leaders in the US and in the world, so it has very good design. In addition to the Mayo Clinic score, which is what the FDA looks at for approval, there are objective measures of the efficacy of bertelimumab. So we're looking at endoscopy. Endoscopy allows to look at mucosal injury as well as histology. This is read centrally in Toronto at the Robarts Institute. Robarts is the gold standard, so anybody that does good trials in IBD goes to Robarts because those guys take out the subjectivity of looking at mucosal injury or histology.
Certainly. So on the surface, having a biomarker that's going to take this very large group of patients and you're going to test into a subset of that patients might seem like you're not getting the full market opportunity, but are there some advantages to targeting these subsets of patients with your biomarker?
Absolutely. When I look at Humira and Remicade, and both are great drugs, the drugs that, beyond the steroids, were the first one to be disease-modifying, which is basically, pushing back surgery. So that's very important. Yet, they only work in about 40 to 50% of the patients. And, physicians and scientists have been looking at ways to identify those responders. And today, I don't think we have really a solution. If you look at the Takeda drug, Entyvio, it's an alpha-4 beta integrin. It works well, and by the way, it works as first-line biologic, even ahead of the anti-TNF, Humira and Remicade. So being more specific, is an advance. Now, Genentech also has an alpha-4 beta integrin, and in order to differentiate themselves from Takeda, what they've done is that they've enriched the population based on a certain number of biomarkers. And although not everything has been published, we know that the results have been incrementally better with the Genentech drug based on patient selection. So what's different about bertoluimab is that eotaxin-1 is not just a biomarker. It's a key regulator of immunoinflammation. It is in the pathophysiology of multiple diseases, including ulcerative colitis, and we can quantify it. In fact, there's an NIH-sponsored study that was published in PLOS ONE in 2013 that looked at 42 different cytokines, so pro-inflammatory proteins, and only eotaxin-1 had correlations with the severity of the disease, whether it's based on the Mayo Clinic score, which we remember is the endpoint for the FDA, or an objective measure such as mucosal injury.
Interesting. So Humira is being given to patients 40–50% who the drug will have no effect in?
Exactly.
And if we can partner a new anti-inflammatory drug with a biomarker, we're only going to be giving that drug in theory to patients that it's going to have a positive effect?
Exactly. So if you look at the NIH-sponsored study that was conducted at Vanderbilt, about 50% of patients had high eotaxin-1, defined as 100 picograms per milligram. We don't know if 100 is the right number versus 80. We'll learn more through the development. But clearly, the concept that if we can treat half of the patients and get a better response, 60%, 70%, 80%, that's really game-changing. And we believe it's the future of medicine. Actually, it's today and going into the future. So personalized medicine, precision medicine, where you give the right drug to the right patients and you get better outcome. It's clearly a huge benefit for the patients first. It's a huge benefit for physicians because they're rewarded by the results. And let's say, it's also a huge reward for the payers because payers, are smart. They don't mind, paying for the right drug, but they're saying, if we have to pay for that drug and it only works in 40% of the patients, we'd rather pay a premium price and it works, in 70 or 80% of 20% or 30% of the patients. So we think that that's the future in oncology, and it's the future in other diseases such as Crohn's and colitis.
Great point, Dan, in that there's been zero pushback on Herceptin. Very expensive cancer drug, but because it's specifically targeted and they're only giving it to these patients, as opposed to some of the controversy we've seen on the pricing of HCV drugs or HCV cures or some of that controversy.
So, we believe that, biology is at the center of the future of medicine. So rather than, going after specific indication and trying to find out, like, in NASH, everybody's trying to see an effect, you have to understand the biology. And, the biology of eotaxin-1, which basically regulates the crosstalk between cytokines and different types of cells, is very important in multiple diseases. So I'll give you an example. We're not like the hundreds of companies that are pursuing NASH blindly, but we have a liver disease advisory board, and that liver docs are pretty close to the gastroenterologist, so that was an easy connection. And it says, guys, we've reviewed the literature, and we see that eotaxin-1 is associated with insulin resistance. We see that neutralizing eotaxin-1 in predictive models reduces inflammation, sometimes even reduces fibrosis. So if we can identify 5% of the NASH patients that overexpress eotaxin-1, maybe your drug will be relevant for those 5 patients as opposed to trying to shoot for 100% of the population. So at Immune
Pharmaceuticals, you're going to using the biology to drive these products forward.
Exactly. And we'll go into clinical trials only when we and our advisors, key opinion leaders, are comfortable that the biology will translate in proper clinical trials.
Well, thank you for coming in today, Dan, and I'm looking forward to 2016 and some of the progress Immune Pharmaceuticals makes.
We're looking forward to 2016, and thanks for the great questions.
My pleasure.